Hello everybody, my name is Luis Fernando Esser, and today I present caretSDM, an R package that unifies species distribution modeling into a single machine learning native and GIS-aware workflow. Species distribution modeling is one of the most active fields in biodiversity informatics, and yet its software landscape remains deeply fragmented. A complete SDM workflow currently demands multiple packages with inconsistent APIs, incompatible data formats, and steep learning curves. And machine learning, on the other hand, in R, has already solved this problem. The caret package unifies over hundreds of algorithms and methods under one consistent interface. Since SDMs draw heavily from machine learning methods, we asked, why should their workflows be any different? caretSDM wraps caret's machine learning engine inside a geoprocessing-aware SDM workflow. It is open source, published on the official repository, and connects occurrence data, environmental predictors, model training, evaluation, and spatial projections into one coherent pipeline. It is extensible for advanced users and yet accessible for beginners, with no compromise on either end. GBIF is a natural starting point of any SDM workflow. caretSDM treats it that way. With a single function, users can obtain data ready to be used in the workflow, running the entire pipeline by concatenating just a few functions. Here are some things that set caretSDM apart from other packages, from which I would highlight its deep GIS and machine learning driven workflow, which allows researchers to spend more time solving biogeographical hypotheses than correcting GIS issues or implementing well-established methods. Let me walk you through a complete example. We start by installing caretSDM from the official repository or from GitHub for the development version. After loading the package, we call these functions to download current and future layers. Here I've put together a function called GBIF modeling, containing all necessary steps for modeling given my methodological choices. We'll be downloading data from GBIF, running a cleaning routine and creating a buffer around records to create a study area. We will run some analysis, retrieve pseudo absences, and train our models using 4 different algorithms. The best models will be projected into future scenarios and assembled. For this example, I used Ilex paraguariensis, the erva mate, with records constrained to the South American continent. The entire workflow completed in a little more than 2 minutes. The resulting object holds everything: occurrences, trained models, performance metrics, and spatial projections, following the ODMAP protocol: the overview, data, model, assessment, and prediction. We can explore results interactively, plotting the occurrences we obtain from GBIF, projections for current scenario, and the projection for future scenario here. Finally, the package is extensively documented. There are 10 vignettes covering multiple topics from beginners to advanced users and benchmarking against other R packages, all available at the package website you can see here. caretSDM is open, reproducible, and built to grow, and I hope it helps you in your next study as well as the whole field. Thank you for your time, and see you in the next video.